Hello, biology class. This is the content video for Unit 2, Section 3. We're going to be talking about how materials are transported into and out of the cell. We're specifically going to pay attention to gases, charged particles, and large particles. So diffusion is one of the main ways that a particle will move into or out of the cell. It is dependent on there being an imbalance of the particle across a membrane. All right, so we need what's called a concentration gradient. All right, so this picture is a great illustration here. On the left side, there is the beginning, and on the right, it's the end. You can see that time progresses linearly, as always. And this is the cell membrane. This is a lipid bilayer. That's not important for you to know right now. Just remember that that is the cell membrane. These blue hexagons are particles. The extracellular space is at the top. Intracellular is at the bottom. So there's a lot of particles up here, but no particles inside the cell. So that is a concentration gradient. As time progresses, some of those particles start to move across the cell membrane down into the intracellular space, the space inside the cell. That continues until we have reached a dynamic equilibrium. Now, a dynamic equilibrium does not mean that movement has stopped. It means that movement continues on with as many particles entering the cell as there are particles entering the outside of the cell or exiting the cell. It's kind of like a seesaw or a teeter-totter, whereas one side goes up, the other side goes down, and vice versa. As soon as a particle enters the cell, another particle is going to be leaving the cell. All right? So there are a couple of different types of diffusion that we need to talk about. There is simple diffusion, which is what we just covered. A lot of times it's just called diffusion. Then there is facilitated diffusion. Facilitated diffusion is where there is a pore or a pump or some kind of actual protein in the cell membrane. That's all these purple spheres or cylinders that allows the passage of a particle into the cell itself. So facilitated diffusion is especially how charged particles, called ions, enter or exit the cell. The big example of that is the sodium-potassium ion pump, and we're going to do something with that a little bit later. There's also the difference between passive transport and active transport. Passive transport, like you see on the left, does not require any energy. It happens on its own. It happens spontaneously. If it's active transport, it requires energy. And the energy of a cell is adenosine triphosphate, that ATP that you see down there below. So with active transport, energy is being consumed to move things across the cell membrane. Sometimes this is with a concentration gradient. Sometimes it is against a concentration gradient. If it is with a concentration gradient, it's diffusion. If it's against, it's not actually diffusion. It's a little different. Don't worry about that. I'm just trying to get that in your head a little. If what's being moved is water, and you're moving across a selectively permeable membrane, like a cell membrane, you have something called osmosis. And osmosis is the same concept as diffusion, but instead of the particles moving, it's the water moving. All right, so you can see on the left side here, this is our beginning state, and over here is the end state. Right, so left is the start, right is the beginning. On the left side, we have an imbalance of water to sugar molecule ratio. Right, so on the left side of this semi-permeable membrane, there's only one sugar molecule, but on the right side, there are 4 sugar molecules. But the water levels are the same. This semi-permeable membrane was going to allow water particles to move from the left side of this beaker to the right side of this beaker. You're going to do an experiment that kind of shows osmosis tomorrow, if all goes according to plan. And here is just a quick Venn diagram of osmosis and diffusion. You can hit pause and read through this on your own. Don't worry about the lowest one on osmosis. All the others are very good things to know and would be a great thing to study. So go ahead and hit pause, read through them on your own. All right. Cellular behavior changes based on what kind of solution it is in, right? Especially when we're referring to water inside the cell. Now, we've got 3 vocab words on this slide alone, so we're going to get to them one at a time. If you have red blood cells in what's called a hypertonic solution, that means there's more water inside the cell than outside the cell. So the water that's inside the cell is going to leave. It's going to osmose across the cell membrane and leave the cell. All right. This is what's being modeled here in this left side. If the water is balanced across the cell membrane, there's just as much water molecules on the outside as there is the inside. We're going to have an isotonic solution. Water will move into the cell just as quickly as it moves out of the cell. That's dynamic equilibrium. If we have a hypotonic solution, there is more water outside the cell than there is water inside the cell, so the water is going to go into the red blood cell. If there's more water inside the red blood cell than the red blood cell can expand to contain, it will rupture. Right? Kind of like if you're blowing up a balloon and you keep pushing air into it, and eventually it pops. All right, so now we've got hypertonic solution, isotonic solution, and hypotonic solution. Make sure you know how water in a cell behaves in each of those types of solutions. All right? If this is a very large particle and it's not going to fit into one of those pores, we're going to have either endocytosis or exocytosis involved in getting it across a plasma membrane. So these are very big particles. Endocytosis sounds like enter. That's exactly what it is. The cell membrane itself absorbs the particles and then pulls them into the inside of the cell as a vesicle. Exocytosis is the exact opposite of that. Exo sounds like exit. That's how I remember those. So if a vesicle is moving towards the plasma membrane, it eventually merges with the plasma membrane itself and expels, pushes out the particles that were inside it, including the fluid that's inside it. So any cytoplasm that was trapped in there with it gets lost as well. I like this diagram because it's color coordinated and it's easy to remember. Endo, enter. Exo, exit. All right, so remember that we talked about how passive transport is one of the main ways that particles can get through the cell membrane, but we may also need active transport as well. We talked about how charged particles are usually going to be using either facilitated diffusion as passive transport or facilitated diffusion as active transport. Right? I can't find, super great diagrams all the time. So remember that this facilitated diffusion, this is also technically facilitated diffusion. It's just active versus passive. Remember we talked about large particles moving in through endocytosis or leaving through exocytosis, and we talked about water movement specifically as osmosis. And its movement depends on the concentration of water outside the cell, either as a hypotonic, an isotonic, or a hypertonic solution. This was a lot of vocab, so make sure you're keeping those words straight. They are all in the Quizlet. So if you need to go review them, go visit Quizlet and review them today. Make sure you've got them down. It may be a good idea to start making your own vocab list as well so that you know which words that you do know and which words you don't know. Of course, if you've joined my Quizlet class like you were supposed to and you have a Quizlet account, I believe that should track it as well. If you have any questions about the material we covered today, go ahead and send me an email. I will help you as soon as I can and get back to you quickly. Have a good day.